All right, today we are going on an incredible journey. We're going to follow a single DNA sample from the moment it's just an invisible molecule in a tube all the way to becoming readable, understandable data. So, have you ever really stopped to wonder how we actually read the code of life? I mean, how do you take something biological, something totally invisible, and turn it into those A's, T's, C's, and G's we can see on a computer screen? Well, guess what? That is exactly the journey we're about to go on together.
The technique we're going to be looking at is called Sanger sequencing. You know, for decades, this thing has been the gold standard. And why? Because it is just incredibly accurate, super reliable, especially when you need to read one specific piece of DNA. It's the classic, tried and true tool that's going to guide our sample through this whole transformation.
Okay, let's do this. Our DNA decoding quest officially begins right now. The sample's ready to go, and its big adventure is about to kick off. So let's meet our hero, this one tiny strand of DNA. But right off the bat, it's got a big problem. There's only one copy, and that is just way, way too small for any machine to even notice. So before we can even think about reading its code, we've got to make a whole lot more of it.
And that brings us to our first major phase, preparation. Seriously, you can think of this like a boot camp for our DNA. We're getting it bulked up and ready for the main event. This whole prep phase breaks down into three really important steps. First, we amplify it. That means making copies. Then we verify. We got to make sure we copied the right thing. And finally, we purify it, which is basically cleaning up all the junk left over. Okay, so that first step is a process you've probably heard of: polymerase chain reaction, or just PCR. The needle in a haystack analogy is perfect here. We're finding that one specific gene we want to study, and then we use this kind of biological copy machine to crank out millions of identical copies. It's like we're building an entire army from just a single soldier. So now that we've got our DNA army, we need to do a quality check. For that, we use something called an agarose gel. This is basically a checkpoint to make sure our copies are all the right size and that we don't have any nasty contamination. We're looking out for annoying little things called primer dimers. That's when the little starter pieces for the reaction just kind of glom onto each other instead of our DNA. If we see a nice, bright, crisp band on that gel, we get the green light. We're good to go. Okay, so we know we have the right DNA, but the mixture it's in is still, well, it's dirty from that whole copying process. It's swimming in leftover primers and a bunch of other unused building blocks. So we use this awesome cleanup kit called ExoSap. It uses enzymes to basically chew up all that junk, leaving us with a totally pure DNA template for what comes next. All right, now we get to the really cool part. This is where the magic happens, for real. In this next step, we're going to tag every single letter of our DNA code, every A, T, C, and G with its own unique color. So what's the secret ingredient for this magic trick? Well, it's these little things called DDNTPs. The easiest way to think about them is as fluorescent stop signs. There's one for each letter, A, T, C, or G, and each one glows with its own special color. So what we do is run another reaction. It's kind of like PCR. And as the new DNA copies are getting built, these fluorescent stop signs get randomly mixed in. And what's the result? You end up with this massive library of DNA fragments of all different lengths. Each fragment stops at a different letter, and importantly, it's tagged with the color of that final letter. But wait, before our colorful fragments can head to the final race, we've got one last cleanup. We got to get rid of any of those leftover fluorescent dyes, we call them dye blobs, that didn't get used. Just a quick purification step, and it makes sure our signal is going to be crystal clear. And with that final cleanup, our DNA fragments are finally ready for the main event, a high-speed, high-stakes race to a laser finish line. This whole process is called capillary electrophoresis. Here's how it works. We load all of our colorful DNA fragments into this super thin tube, a capillary. Then we apply an electric charge, which starts pulling them through. Now it becomes a race. The smaller, lighter fragments zip ahead, and the bigger, heavier ones lag behind. And right at the finish line, there's a laser, and it reads the unique color of every single fragment as it crosses. Okay, the race is over, but we're not quite done. We don't have our sequence, not yet. Now comes the translation part. We have to turn all those flashing colors from the laser into an actual readable code. This is what the raw data from the laser looks like. It's called an electrophoregram, which is a fancy name for a chart with a bunch of colorful peaks. Each one of those peaks is a different DNA fragment crossing that finish line. It's pretty simple, actually. The color of the peak tells you the letter, and the order they appear in tells you the sequence. Cool, right? And here it is, the final translation. The computer software takes all those colorful peaks and converts them into the most basic, universal format for a DNA sequence, a text file called a FASTA format. It's really just a simple header with some info, followed by that long string of letters. So, we have it, our sequence. We've successfully read the DNA. But there's one last, crucial question we need to answer. What does this code actually belong to? To figure that out, we turn to an amazing online tool called BLAST. Think of it like a search engine for DNA. We literally just copy our FASTA sequence and paste it into this huge global database. BLAST then scans everything and compares our sequence against all known sequences in the world to confirm what it is and even spot if there are any interesting mutations or variations. And that's it. We did it. That really is the full journey, from a simple tube of DNA all the way to a confirmed sequence. From a molecule you can't even see to a string of code that's now verified and part of a global database. Pretty amazing. You know, when you think about it, it's kind of mind-blowing. This process lets us read the most fundamental code of life itself with stunning precision, which really leaves us all with a pretty big, fascinating question to chew on. Now that we can read the code, what secrets are we going to choose to unlock next?